okay? But I'm going to walk you through meiosis, okay? So this is an organism that has, let's see how many pairs. We've got 1, 2, count them with me, 3, 4, 5, 6, 7, 8, 9 pairs. So how many total chromosomes for this one? 18, okay? Now, we are already assuming that replication has taken place, okay? So, in actuality, this one has made a copy of itself, this one has made a copy of itself, and since these are both chromosome 4, okay, then they have met together. Got it? All right, so we're in prophase 1 right now, okay, because they're still kind of jumbled up in the nucleus, but all the tetrads have formed because the homologous chromosomes have come together. In this case, I was lucky enough to get red ones on each, so we're going to say the red ones are from the mother, okay? And so all these red ones are from the mom, and the other colors are from dad. Got it? So chromosome 1 again, and you can count all the way down to chromosome 8. Got it? 9, sorry, 9, yeah, 18 total. Okay, so one of the first things that can happen in prophase 1 is crossing over, okay? So y'all are gonna all get a chance to do this, but I'm gonna slide some out, and you can share them with 2 people around you, okay? All right, so slide them out. We're still in the nucleus here. And so with crossing over, one of the things that can happen is that 2 of these legs of the chromosome can cross over, all right? And because they carried similar genes on them, they can actually swap places. You know when they cut jigsaw puzzles, they use this huge machine that just cuts it. And the picture changes every time it goes onto that big machine. But if I count over 6 and down 9 and take that piece, it's going to fit in the next picture. Even if the illustration doesn't match, the piece is going to fit, right? So these genes are close enough in DNA structure that they can swap over, all right? So I'm going to count off 4 here, and I'm going to count off 4 here. It has to be an even number, and I'm going to swap the yellow on the red and the red on the yellow to simulate crossing over, and you do the same to your bottom 2, okay? And it doesn't have to be 4. You can choose 2, 3, 5, 1, okay? It's up to you. And then we're going to cross over the inside pair at the top, all right? And this time I'm gonna take a different number, okay? And I'm gonna cross them over, and you can let somebody else do it, okay?
Make sure you do the other half.
You don't have to, because crossing over doesn't always happen. So you don't have to always cross over. Got it? Okay, so we're finishing with prophase 1. All right, what happened with your pair? Oh, you got it there. Okay, it should be, I think, like this. Okay, flip the other one. Yeah, we just, since y'all did that. Okay, so we flipped them over, and we've got some. Just make sure that all your ones that crossed over on the right sides. Yeah, does that one keep coming out? Okay. All right, so let's go into metaphase 1. So in metaphase 1, the tetrads are going to stay together, and we're going to all line up in a single row down the equator of the nucleus. So line up. Doesn't matter what side you put the red one on, okay? Okay, so here is metaphase 1. Notice instead of 1 line down the center of the nucleus, okay, or what was the nucleus, we now have 2, because there's 2 sets of chromosomes down the middle. So that's a difference with meiosis compared to mitosis. Got it? All right, so this is metaphase 1. So crossing over happened. I call that cutting the deck. You know how when you play cards to make sure you get a different roll, a different deal the next time, you cut the deck and you shuffle, right? So the first thing that happened was crossing over. That's cutting the deck, all right? So that we don't just get red or black. In this case, we don't just give mom or dad's chromosomes, we give our own mix, all right? Here's the next pair, the next set, and this is independent assortment. You notice that the first 4 are mom's chromosomes on the left here and dad's on the right, but then we swap here, we swap back, we swap here, and then we have 2 more. So if this child were to be fertilized by a sperm, they might have a lot of mom's genes, and you might have a child that looks more like mom than like dad. If it was the other half, they would look more like dad maybe than like mom. Got it? All right, so that's how siblings can be very different from each other, even though they have the same parent. Okay, we're going to anaphase one, so I need some hands to help me, okay? So we're going to split the homologous pairs, all right? We're going to split the homologous pairs into 2 cells. Keep going, all the way back, pull them back, okay? All right, so now then we're going to have cytokinesis, right? Cytokinesis, and these will clump back together into a nucleus like what happens in telophase. All right, so how many cells do we have at this point?
2.
How many cells do we have at this point?
2.
2, okay? Because cytokinesis has happened. So we have 2 cells at this point, okay? Now, but how many copies of the entire set of DNA do each of them have? Yeah, they only have 1 at this point. So this is where you can get in the argument as to whether they're already haploid or if they're diploid at this point, okay? So it depends on which book you find, and they're not going to ask you that at the biology 1 level, okay? So you can, if you want to write an essay and argue about it, you're welcome to. Okay, so now I'm going to send this one over here, okay? And I'm going to send this one over here. Whoops, we lost the tail there, okay? All right, so I want y'all to keep them together with their partners and line them up again. So that was prophase 2. Line them up again now for metaphase 2. Metaphase 2, okay? We might have to curve our line down the equator, okay? So now we have metaphase 2. Line them up. It's okay. Line them up. Line them up. Okay? There we go. Make a line. Okay, make a line, metaphase 2. There we go. Your line has to curve because of our table size, okay? All right, so we've got metaphase 2. All right, they're lined up in the middle. And now let's go into anaphase 2. You ready? So try to keep the inside stays together and the outside stays together, okay? Help. Come on, because I can't reach. Help. All right? So all the inside ones come here. And the outside ones come there. Okay. All right, and those are the the outsides. Put those together. All right, now how many daughter cells have we formed?
4.
4. How many chromosomes do they have compared to the initial set? You counted them. How many?
9.
9. And they started with?
18.
18. They had 9 pairs, but they started with 18. Now they're no longer pairs. They're just single chromatids, right? Okay, so if this becomes an egg cell, all right, then where is it going to get the other half set of its chromosomes?
The other daughter cell.
If this is an egg cell, where is it going to get the other half of its chromosomes? From the sperm cell that fertilizes it. That's right. So whatever sperm cell comes in, whatever combination it has from the father's parents, are going to combine with these to make a own unique combination, okay? Now, for a single daughter cell, it's 2 to the 23rd, okay? So 2 to the power of 23 for the possible combinations, okay?
That's a
And
big
then
number.
you multiply that times the dad's combinations. You know how they say you're 1 in a million? You're actually 1 in about 8.3 million. Okay? So pretty cool, huh? All right? That's all. That's right. That you can tell them why, okay? Because that's all the possible combinations. And then you add crossing over, and that number goes up even higher. Okay? All right. So, again, did we get more red ones? Look at this one. Mostly mom's genes there, okay? And look, this one's got lots of dads, and that one has kind of a half and half. All right, so they shuffled again during metaphase 2 as they lined up, okay? So just like when you play cards, cut the deck, shuffle, shuffle, okay? All right, so cut the deck in prophase 1, shuffle at meta and ana as they separate, and shuffle again at meta ana 2. Got it?
We should do it again.
Okay, I think once